So thank you very much, Martin, and Anna, and Kevin as well, for inviting me here. I'm coming from a scientific part of the discussion. That means I'm a chemist. I studied chemistry. I do my own research. And I was lucky enough that I worked in Karlsruhe, where I got in touch with the Center for Media and Art. So I used to work with some of the artists, and we engage in discussion. How can we actually do things together? How can we communicate? So we also kick-started a symposium which is called Molecular Aesthetics. And this was also very inspirational for me, because I always believed that art and scientists are, of course, very connected, and I will show you something what I think is important to see, and I actually got lots of inspiration from it as well. So I'll tell you a little bit about seeing the invisible. I work in nanosciences, and so we have lots of microscopes, lots of data that we need to interpret, lots of pictures that we need to enhance to actually be able to see what is the scientific background of it. So let me see. Okay, and when we talk about, I need to switch this on. Okay, but here's something for the introduction, what I would like to talk to you. When we talk about aesthetics, and I will start a little bit about molecular aesthetics, because this is something that I was working on with Peter Weibel, the artist. For long, the aesthetic was considered to be a perception of beautiful. It was fine arts. It was something which was perceived as beautiful, and then also it became a theory of this beautiful, the theory of fine arts. But today, the enhancement of this definition is basically needed, because we have today a general perception, and we have a cognition enhancements. Because of all these microscopes we have, because of all technological means, we actually can go beyond our sensory feelings, because we can go beyond what our eyes can see, what we can hear, because we can enhance many things. So we have this enhanced sensory cognition. We enhance it by use of media. So how did our perception of aesthetics and fine arts actually change as well? So this is the question. And actually one of the first guys that mentioned the term molecular aesthetics was a chemist, a German chemist, Professor Roth. And he wrote this little book in the 70s already, and it was only published in German, where he talked about the perception of molecules, about the symmetries, about how the beauty of a molecule enhances our our attraction to it. Do we actually find it exciting? Do we find it not exciting? And he talked, this was like a book, it's a 30 pages book, which was really interesting because it put a new concept of a chemistry and the symmetry, thank you, onto it. And then Peter Weibel, who's the director of a Center for Art and Media, also wrote several essays, and he put this new term out there, which is called Renaissance 2.0. It's a new age of aesthetics, which is expanded by non-sensory and artificial perception. That means everything that we use to enhance our sight, to enhance the things that we hear, the things that we feel. So molecular aesthetics and this Renaissance 2.0 concept, and there will be an exhibition in June 2015, which is called Renaissance 2.0, which will actually join artists and scientists together. So it's a merging of art and science to expand our experience of both. And so, when I was thinking about what scientists and what artists think, because, we analyze, the scientists always need some kind of little bit of a definition. Scientists explore and open new realities. That means we explore something that was maybe not known any longer till then, and in this way we open new realities, we open new fields, we discover something new that can start up a new field. Artists also create new realities, because every piece of artwork is something new, something that came individually and uniquely from an artist. And of course both groups can have mutual benefits. One thing that is also very important before I show you some examples is that reality is now moved from the observer. from observer relative to observer constructed. That means we are just not the people that observe and describe. We actually construct now some technical means, some instruments by which we can create new realities. And this is, for example, one picture that I want to show you of that always impressed me. You know, as a scientist, you are always faced with some certain level of frustration, because you can work for 6 months, 1 year on the project without any single result. And you need to have the motivation to work a little bit more. So, and not to give up. And this is one of the pictures that I like to look. This is done with the electron microscope. And what you can actually see is basically the atoms of a silver in a very small silver nanoparticle. So thanks to electron microscope, we moved from this theoretical concept, where actually Niels Bohr, who used to live in Copenhagen, was very important in putting the whole theory of atoms in place. So thanks to the microscope, we see something that once considered a purely theoretical concept. So we can see them. These little dots are actually the atoms of a silver. So this is, for example, always exciting for me, because it makes me feel like, wow, I can see the things that I used to read about before. So, molecular aesthetics, and these are 3 questions that I will try to discuss throughout my talk, and maybe give you or offer you some answers, and we can discuss that. How did being able really to see down the atomic level, to see something that we couldn't have seen before this age of new technological developments, how did that change how we perceive the nature and the world around us? How did that change how artists see the world around us? What role was played by the color and super resolution microscopes, for example, throughout these developments? Can we, also inspired by molecular aesthetics, become more sensitized to the beauty around us? You know, because now it's not only about seeing what is there, what we can see with the eyes, we can also actually see beyond what matter is made of. So something which is purely black could actually have more meaning because it's made of a certain nanomaterial, which takes years to develop. And, you know, are we going to be able to bring into life new art forms, create new nature? And some of the questions concerning molecular aesthetics, I could definitely recommend the introductory essay by Peter Weibel in our book, where I think he very nicely summarized some of the developments. So this is how the journey is going to look like. We will explore a little bit the changes that technological advances brought to our perception. We will explore molecules and artworks which were stemming from the molecules. That means artists that were really directly inspired by molecular world. And we will try to look into the real example of art is life and life is art. So as a chemist, I will start with some of the molecules. And I told you already that the matter is made of atoms. And physicists, of course, would not stop there. He would also say that there are some subatomic particles. But we will focus on the matters. And Peter Stagely, for example, he also, one of his artworks was this video rotor, which actually describes very nicely how the atom looks like. Because atom has the nucleus and it has the electrons, which are constantly moving around this nucleus. So basically, when you would look at the atom, it would more look like this than like this. That means the artist feeling about the things is actually very precisely and very close to the scientific description, to the quantum mechanics of it. This is how the molecules look like for the non-scientists. One of my favorite molecules, of course, water. Not for the reason that it's so important for our life, but because it's really a very exotic molecule from the chemical point of view. You will be surprised. I was surprised. And of course, then you can have a very complex molecule. This is, for example, B12 vitamin, which was also very important for a range of discoveries in chemistry. So, as a chemist, we use symbols to represent the molecules. And symbols are letters and numbers. But this is also very limiting, because you can have a molecule that has the same symbols, the same numbers, but it has a different structures and different properties as well. So we needed to develop a whole new language within chemistry that will help us to describe the molecules as well. So, for example, the symmetry determines the function. So we have, for example, the whole charts, sub-naming of the molecules. It can be very confusing sometimes. And since the symmetry also determines the function, not only the number and the type of the atoms of the molecules, this is, for example, one of the examples of Contagan, a very dangerous molecule that in the 60s was prescribed as a sleep medication, and it caused lots of deformation in children as well. And it has, you know, the same structure. It's just oriented in space a little bit differently. So symmetry is a little bit different. One form is extremely toxic, another one is not. That means in chemistry, the symmetry also plays important role. And I think this was also something which was fascinating to artists. How can the things have such a different properties just based on the symmetric structure? And since there is a huge number of molecules, of course, and the atoms and elements, chemical elements which we needed, as a chemist, we needed some kind of orientation system, some kind of coordination system. And this was for us periodic table, which was first time put to life with Dmitry Mendeleev in 19th century. So he was the first one that selected, that kind of put the elements in order. And this is fascinating. And he actually had a circle of friends who were artists in Russia. And he sometimes also said in the interviews that he was also driven by these discussions that he had with them to think about, to think in a different way. He was inspired to see the things that were in front of him differently. I mean, lots of chemists knew that there are so many elements, but nobody thought about, hey, how can I put them all together and what kind of relation they are? So, of course, something, these discussions with his friends also helped me. So, we got this periodic table, which is where the elements are sorted out in groups, and you can predict the properties of it. The periodic table was, of course, also inspiration for a few artists. Blair Bradshaw, for example, did a lot of periodic table variation, oil on canvas, which is always very interesting for us to see as well. Molecules of life. We are fascinated by this as a chemist, as a scientist, but also lots of artists were fascinated about this. When we talk about molecules of life, these are the molecules that build our body, that build the nature. We are talking about DNA, which you, of course, recognize, because this is really one of the most often. I did a little bit of research on DNA and how often is this structure used in marketing, for example, and you would be surprised what kind of products you can basically, you know, do the marketing with using this kind of helical structure. You have proteins, which are very important elements in controlling our metabolism, growth. This is a hemoglobin, which is important for oxygen binding. Without this, we wouldn't be able to transport the oxygen. And they all have amazingly wonderful structures. These are antibodies which help us to fight the colds. For example, I'm definitely having lots of them now in my body. And they have this interesting structure. Molecules and molecules of life are tiny. And usually we say they are on 1 nanometer scale, so there are lots of zeros before here, before the meter. And for example, this antibody structure was inspiration for Julien Vos-André, who is a very interesting guy, and I actually had a chance to talk to him. He used lots of molecules as inspiration for his artworks. So he superimposed the structure of antibody, so he enhanced the scale, and he saw that the symmetry of the antibody corresponds to the symmetry of a Leonardo's Vitruvian man, which is also very interesting. So in 15th century, Leonardo actually proposed or established a mission statement of the classical aesthetics, that this represents shapes of all visible objects. That means you see something, you recognize the shape. This is the definition of a classical aesthetics. In 20th century, we needed changes to this mission statement because we now see the molecules and a new reality is formed because we have all of these molecules make us. They make the matter around us. So there is a new definition of aesthetics which we need. So this is, for example, the artist's perception of the things. So hemoglobin, the oxygen binding molecule, was also inspiration for Arvin Guy's. This is the cytochrome C, sorry, this is a very similar protein to the hemoglobin because it has a center and it has several molecules, several atoms around it. So this was inspiration for some of the artists because of the beauty of it and the symmetry of the whole molecular structure. This is, for example, Drew Berry is an Australian biologist who went into the visual arts. So he's a very well-known also in scientific circles. Whenever we need something for a cover of a magazine or for a really beautiful presentation, you would like to contact Drew Berry because he has a very imaginative way of dealing with the molecules. He really works on the interface of art and science. So he precisely gives you the message which we as a scientist can accept because it's scientifically correct and still puts a little bit of an artistic interpretation in it. Joe Davis as well, he works a lot with the DNA structure. He's fascinated by DNA structures but also by information which is contained in the DNA. DNA is almost like a small computer that has a set of data and can transfer the information to the throughout the cell to produce the molecules. So of course some of the artists are fascinated by this, and Joe, for example, writes coded messages in DNA and sends them to space. It's really interesting as well, or put them in the bacteria. So this is a new level of molecular engineering done by artists. Then of course, we also in my group, I always try to encourage my students to think beyond the scientific experiments as well, to play a little bit. I think this playing, this romanticism that Martin actually mentioned is very important also for a scientific discovery. You need to keep the sense of a game, the sense of excitement that you might be discovering something new. So we play with our images as well. So this is what we made for the Royal Society of Chemistry, where we took really real life images done with a microscope of different nanomaterials, just change a little bit of the colors, and did something which we called Nano Warhol. So, sorry. Going back to molecular aesthetics and to Julian, again, and the hemoglobin, he did one interesting sculpture. And he did a sculpture of hemoglobin. Here in the center, you can see the iron center, which usually binds oxygen, and then you have a protein structure. If you leave this exposed to the elements, it will oxidize in the air. It will bind the oxygen. This is exactly what protein does. It binds the oxygen, and Julian transferred that in the real life experiment, artistic experiments, which you have a sculpture which stays in interaction for 3 months with the oxygen, and it changes color. So you can basically feel this interaction that is happening. So saturation of oxygen is brought upon the changes in the sculptures. The interesting thing that also happens, and was inspired by molecular science, is a psychedelic art, which was kick-started by LSD generation. That means lots of artists who took LSD. This is the LSD molecule, quite fascinating as well, very difficult to synthesize in the lab. And for example, these are some of the examples from the movie, from the film stills, 60s, the examples of psychedelic art, Gerd Stern. This is a real-life image of a liquid crystal which is put between 2 glass plates. And this is fascinating to watch. You can do these experiments in the lab and you can do some microscopic images. So you get these wonderful, amazing images in different colors, which really reminds you on the works of a psychedelic artist in 60s. So this is where, you know, even without knowing, the artist created something that exists in nature already, we were just not able to see it. So the beginnings, a little bit is very important, I think, just to see how the microscopy, how the seeing and invisible actually happened. And microscopes were developed in the 16th century. And Robert Hooke was one of the first that used them to look at the living cells. And he said, by the help of microscopes, there is nothing so small to escape our inquiry. Of course, he was not right. But it was a huge achievement. And he actually looked at the cells of a bottle cork, and he defined cells, because he called them, they look like little cells where monks used to live, cellule. And he said, okay, I'm going to call these structures cells. So he defined what we today call cells. And then lots of scientists at that time, at the beginning of modern science, were amazing artists as well. They were excellent in drawing. And this, of course, helped preserving some of the discoveries. Leeuwenhoek, who was exploring the cells, drew some excellent images of spermatozoa, sperm cells, which are totally, if you would look in the school books today, absolutely right. So he got it totally right because he had wonderful observational capabilities, and he also drew very nicely. So when Hooke saw the cells, and he said nothing will escape our perception any longer, and he usually looked also at the brain cells, he would look from the dogs, and he saw only gray masses, and he could recognize certain kind of structures. But then came the color. Then scientists took the inspiration and used some of the colors that artists have been mixing and simply put the cells in it, trying to see what happens. I still believe, you know, historically, you never know how they started. I still believe that there is lots of serendipity. Maybe they dropped a little cell in the color and you know, just wanted to check. And then they realized this is helpful. Usually this is how big discoveries also happen. So this was how, for example, brain tissue was drawn at that time, when Joseph von Gerlach used carmine, a red color that was used in textile industry. So he could see the structures. This is how we see the brain cells today. So he was not very wrong at that time. Camillo Golgi is one of the scientists who used silver staining. That means the same thing that was used in the photography, to see intricate things in our neurons. So that means the colors were binding differently to the different organelles. So suddenly people could see different parts of the cells. Today, of course, we have fluorescent colors that can really show you different parts of the cell. So for example, here you can see the membrane of the cell. Here you can see the nucleus. So colors brought another reality to these cells, which used to be a gray mass, but now not any longer. Now you can really see what is in the cell, and you expanded that knowledge. One thing that really brought a revolution to molecular science was the discovery of fluorescent proteins, particular proteins that have colors, which are enhanced under the irradiation. And this was beginning of 90s, that means not so long ago. And yet from the beginning of 90s till today, there were really significant advances in molecular biology. And these fluorescent proteins were isolated from nature. There are aquatic organisms like this Aquaria victoria, which usually use these proteins to communicate, and also to, basically to communicate, to send the signals. And a group of scientists isolated them. They have remarkable structures because they have this fluorescent color inside, and they have this protein coat around them to protect the color, so nothing can basically happen to it. And of course you can isolate many of them molecularly, you can make different colors. And this is what the artists in San Diego, not the artists, but the scientists in San Diego University used. They used bacteria which had 8 different proteins, 8 different colors, and then they drew this picture of a palm tree and the dolphin, trying to make the art of it. And if you look at it, this, you wouldn't say this is necessarily art. You could say that this was just somebody has tried to do something where you can recognize certain elements. But if you think that these are bacteria where you needed to engineer 8 different colors just to make that on the certain agar plate, then it gets another significance to it. The use of fluorescent colors, of course, and fluorescent microscope, which was developed at the end of the 19th century, but now in the 20th century we have a super resolution microscopes. This enabled us to see extreme details of the cells. For example, here is the division of 2 cells, and you can see exactly the moments of this division, how this happens. You could see, for example, neurons, how they look like. They are pyramidal. And of course, the more you see, the more you can understand the function of it. You can postulate the function and you can develop some of the stuff. And then the artists come in, and Overgaden, he's a neurobiologist who really liked Japanese art. And he thought, hey, sometimes when I observe these things in the brain, they look so amazingly beautiful. Can I do something about that? And he developed a new methodology where he uses etching in gold. So he uses lasers to etch the things in the gold, and he had the whole set of artwork which is inspired by brain cells, by neurons, by something that he saw. But he puts a new technique, which is a laser etching of the gold, and he puts his own interpretation of this beauty that he saw under the microscope. And when we talk about microscope, and I know there will be talk later on, but I will just briefly go, because they enable us to see invisible. Optical microscopes of that kind that Robert Hooke developed in the 16th century, the best one had a resolution to 0.2 micrometers. That means you could see the objects of that size very nicely. And if you look at this scale, you could see the hand, hair, and sometimes a bigger cell, and E. coli, which is a bacteria. You could also see cell parts. But if you go beyond that, you get quite a smaller scale, which you can't see with the optical microscope. So still, until the development of super resolution, all of these things were hidden from us. We didn't know how they looked like. We knew partially because we used different techniques, but we couldn't visualize them. So viruses are much smaller, proteins, and then you have small molecules which are 1 nanometer big. And this is what is interesting. This year's Nobel Prize was given exactly to the people who pushed the resolution of these microscopes, who enable us to see the nano world, to see these parts which are 10 to 1 nanometer. And this is called stimulated depletion emission, the physics behind that. We will not get into that now, but the important thing is that resolution was extremely improved. So now you just didn't see the parts of the cells or the cell membrane as I showed you, but now you could see the individual molecules. That means you can follow, you still need to do certain things. It's not so straightforward, so you still need to chemically sometimes modify the molecules, but you could basically track a single molecule within the cell. And you can imagine that this is also beyond something that we as a scientist considered invisible. So quite rightly the Nobel Prize was awarded to them this year. And I wanted to show you 2 examples, 2 real-life examples from some of my collaborators. And here you have living neuron, where you basically see how it fires these little hotspots of a light on the neuron. Is when the neuron fires electrical signal, when it communicates, when it does something. And also here, I will see if I can play that. This is, I asked one of my collaborators from Italy to send me this. Basically, what you do, you label certain molecules with fluorophores. And this is also, this is the neuron. And you can follow the movement of these molecules along the neuronal cells. And if you think that the memory, you know, our inspiration, creativity, is all stored in the brain, it's all based on this electrical communication between our neurons, and now we are really capable to follow these electrical signals. I mean, we don't understand nothing yet. You know, we just scratch the surface. But thanks to the microscopes and new chemical methods, we could actually, now we started to follow these impulses through the neurons, which is quite amazing. And when we talk about visualization in science and what we have taken from art, of course, we need to go back to the colors. And you know, when we think about colors, we know that these are some kind of molecules that have certain properties. So, for example, one of the most often used colors in industry is this pigment red, 170. So it has this kind of structure, it's a red powder. But when you look closely, you could see that this is a crystal structure of it. So it has layers of a crystalline structures. If you analyze that, then you will see that there are certain molecular structures which are connected, and this is finally one molecular unit of this pigment red. So you start with this red thing, and then you do the crystal structure, then you understand a little bit more, and then you totally understand what this color is made of. So some of my friends would say, but then you lose this romantic thing, you know? You know what is there, and you see that this is this molecule. What is so exciting about it? But I think exactly then the excitement starts, because you can change the things here. So if you change something here, you will change the property of this. So this is also something very exciting. But you have also molecules which are important and in nature, which are very difficult to synthesize in the lab, like a chlorophyll that has this kind of structure and gives the green color to the leaf. Without this, we wouldn't be living today, because we wouldn't have oxygen, for example, and lots of food. So this is this question, knowing the structure of the color of something that is really beautiful, that destroy the aesthetic experience. And the Kantian view would be an object that is of utility, whose valuation is not sensually immediate but requires cognitive action, cannot qualify as beautiful. But for me, it's totally different. The more I know about it, the more I know this beautiful molecular structure behind it, it actually enhances my aesthetic feeling of it. So this is in this new dimension of aesthetics, is understanding and utility. But you know, colors is not only about molecules that give you certain kind of property. There is something which is called structural color. So that means you can have certain molecules, and if you change how they are positioned in the space, are they parallel to each other or horizontal, you can change the way the light from outside interacts with them. So you also change the color. So basically, you know, if you have a blue eyes, if you have a green eyes, if you have a brown eye, it's not a different color that you have in that, it's not a different molecule you have in your eyes. It's the same molecule, which is just, and this is this, melanin, and there is also iris pigment. And the position of this molecule in space determines what kind of color you will have. So we all have the same one, it's just positioned a little bit differently. These structural colors are of course very important now for nanoscience, because for example, if you look at the birds, they also don't have a certain molecule that determines blue color. By the way, blue color is very rare also for chemists. And in the nature, it's very difficult to make blue colors. Very interesting. Maybe there is a planet somewhere in our galaxy where blue color is not rare, but it's not Earth. So this is how the bird's feathers look like. And this is microscopic image of a bird's feather. And it's basically sponges filled with air bubbles. And because you have these sponges which are filled with air bubbles, light behaves differently when it interacts, this interface of this organic material and air, and that's why you get the blue color. So there is not a molecule that determines that. It's basically a physical interaction that gives the blue color to the wings. And this is where nanomaterials get their importance as well, because they are very small assemblies of atoms. They can be made of silver, gold, they can be made of any other metals, for example. And they have interesting properties. The smaller they get, the more interesting sometimes they are. The gold necklace that you have will have totally different properties if it's made of gold nanoparticles. And color in the nano world depends on the size, not the structure of the molecules. And some of the artists have been also using this in the recent work. So when we talk about visualization and the rise of nano, just for the comparison, sometimes it's very difficult also for me to imagine what does it mean, nano? It's so small, you know, you have so many zeros before this one in the meter, I can't imagine it. But this is one analogy that one of the scientists that I like use. And if you take a nanoparticle, which is 10 nanometers, these nanoparticles to the football is the same as the football would be to the whole Earth, which means this nanoparticle is very small. And this is not a new phenomenon. You know, maybe sometimes you read in a newspaper and everybody's talking about nano and this and that. It's nothing new. It existed already for centuries. For example, Romans have used nanoparticles to get a red color of a certain glass. They stained, some of the stained church windows are made with nanoparticles. The thing is, people didn't know what is in it. So they developed this process. They realized, oh, I get a nice yellow color, I get a nice red color. But nobody had the ability to analyze what is that. And we now know that this is nothing but the nanoparticles that give the color to the glass. Or the wonderful swords that used to be made in Syria, in Damascus, which are really famous in 15th century because they were so sharp. We now know that they have nanotubes on their surface because they were prepared in such a way that nanotubes were grown on the surface and they gave them the sharpness. So this is all very interesting, you know, the things are around them, but we can't really see them, so we don't analyze them. So this is just to tell you now, to close up a little bit, this is how the microscopes today look like, this is how they look like before. So you still recognize the tubes, but they look a little bit different. You have also the computer. What you get now, you know, when you look to the optical microscope, and you probably did that in your biology class, you look through the sample and you see the sample. You can draw it. In new electronic microscopes, you get a set of data. And you need to analyze, you allow computer to analyze this data, and then you process the data, and then you get a picture. So this is how it looks like. For example, this is atomic force microscopy, where you can check the cells, how they look like. So you have this little kind of probe, which almost touches the cells. What you get is a set of data like this. These are the numbers you get from this. This doesn't tell you anything about the cell, how it looks like. So you have computer programs which will transfer this data into this kind of image. And then if you want to enhance it and you want to see which part of a cell is higher, which one is deeper, what is the three-dimensional structure of a cell, then you add a little bit of a false color. This is all done in the computer. So you can ask yourself, is that real? Maybe the machine is lying. So, is that real? Well, I hope yes. I hope there are not some kind of strange aliens that are really destroying all our instruments and so we get a false image. But this is what you get. I was also amazed for the first time when I saw the type of the data. Let's imagine in the future you really get files and files of this. You wouldn't know what that means if you didn't have a software for the interpretation. It's a bit scary. So seeing the invisible, so from this data you can see really the structures, assembly of nanorods. This looks like a really some kind of strange cannon. Or you can even write the things with the help of atoms. This is IBM. This was a special technology which you can use the atoms to position certain things. But then again, you can see the invisible in nature. For example, this is the moth eye. Or the wings, how they look like. So the wings are basically horizontal, the vertical positions of particular molecular structures. And this is, for example, how the structures of a certain plant leaves look like if you look at them with the microscopes as well. Or maybe the butterfly wings. When you go on towards the nanoscale, you could see the intricate structures. And this is, for example, what now the scientists are using to create new materials. So you can use, when you understand what this is made of and why is it so colorful and strong, then you can design the materials which are based on this. Or the lotus, which is now used for many materials in the car, so because they are water repellent. It's called the lotus effect, and it was used to design new materials which repel water. So this is all possible in chemistry. And now, one thing which might be a little bit scary, when I told you we could create new nature, so I asked a friend of mine who's a physicist, and they develop a new technique where they can write with electron beam. They can write certain things. So they took a mosquito, which was dead already, from the lab. And you know, mosquito have, they have this little stingy thing which they use to sting. And look at this now. So they wrote, if you look at this and enhance it a little bit, you see the tip on this tip of a mosquito. And if you look at the structure a little bit more, you see a little bit more defined structure of this tip here. If you go closer on the tip of the tip, you will see that there is something written. And this is CFN, which is the name of the institute where they work. So imagine the scale of the things. Imagine how many things you can do on that scale. You will never see it. It's a little bit scary, but still possible to do with the techniques that we do today. And finally, I will close up with the symmetry, because when I talk about molecular aesthetics and I talk about microscopes so that we can see what we can actually do with them and the colors, how this is done and enhanced. But also one important thing for us scientists and also for artists is the symmetry. And these are two fascinating things that are very interesting to me. This is dark matter, and dark matter should be everywhere in the universe. It's a very interesting physical concept. And this is the first map of a dark matter, which was published this year. And this is how it looks like, just the scale is different. If you look at the gray matter, and this is the brain, it looks pretty much similar to that. I'm still very amazed that there are certain patterns in the nature. I'm sorry. And this is artist matter, I call it. This is the work of Greg. And now we have X-ray crystallography, which told us a little bit more about molecular symmetry. And this is what you would do when you do X-ray crystallography. That means you are exploring the structure of the crystals. This is the image you will get. It doesn't tell you much. If you are not a crystallographer, I don't know, I wouldn't be able to interpret that. I would just be able to say that it's symmetrical. This side is symmetrical to the others. And this is what was called one of the most beautiful X-ray photographs of any substance taken. And this substance is DNA. So without this image, which was done by Rosalind Franklin, this wonderful resolution of the image, the Watson and Crick wouldn't be able to propose the DNA structure. I mean, somebody would eventually, but it wouldn't be done if she didn't do such a nice image. And I'm sure that molecular biology would be stale for several years. So this is how the symmetry looks like in DNA, in our molecules. I will not go to the details. And this is what we do with the same DNA today in science. We develop a technique where you can take a viral DNA and you can fold it in different shapes. You use the computer programs to design the shapes, and then you can use certain kind of parts of a DNA to make the folding. And these are the real microscopic images. So you can make smileys, you can make different shapes, you can even make this kind of vessels. Everything made of DNA, everything on a nanoscale. And you probably ask yourself, why is it that interesting? First of all, whole 4 years, nobody knew what can we do with this, apart that it's being very nice because you can manipulate the molecules. Now, of course, we understand, and people are working on this, using these vessels to transport certain things in the cells. You know, because DNA is it's a biomolecule, so you can, it's friendly to the cells. Or you can maybe use this kind of cubes to put certain drug inside and transport it through the cell and open it up. So, you know, first it was a purely, let's see and let's play and let's see what we can do. And now, of course, we are discovering ways of using this. It's called DNA origami. Talking about crystals, of course, crystals are very fascinating to the scientists. So Ljiljana Fruk, for example, had a whole range of artworks which were based on 3D animations of crystal growths. Very interesting work as well. Some of the works, some of the molecular structures were first done in architecture. For example, Fuller Wrench. This is the original sketch by Buckminster Fuller, who was a famous architect in 1954. And this is the structure of a Fuller Wrench molecule. And without this work of a Fuller, it wouldn't be the Harold Kroto and Richard Smiley wouldn't have guessed the structure of this molecule, because we all believed in chemistry that something like this is impossible. It's 60 carbon atoms arranged in this wonderfully symmetrical arrangement. And they wondered really for a long time, what could that be? And architecture actually inspired an understanding of a discovery. The same was with the nanotubes. This is the model that Fuller made. He played with the symmetry and made this model. If you look at the structure of a carbon nanotubes from 91, this is exactly the structure you are getting. Of course, the symmetry was present in art already before. I'm collaborating with Martin Cowling, who's a Chilean artist, very much interested in the symmetry and in hexagons. So he went all over Europe looking for architectures, and that could remind him on a molecular symmetry. So in Topkapi Palace in Istanbul, he found this. If you look at the structure of graphene, you see the similarities. So there are certain symmetrical patterns that repeat. Water fountain in Morocco looks exactly the same as a boron compound, which is crystallized. Church window, I think this was somewhere in England, resembles a metal complex done by Leonard Lindoy, which is a famous Australian artist. So, you know, there is a symmetry all over us, and even without seeing the molecular symmetry, people and architects were able to put the mathematical principle into work. And this is one of the patterns in Morocco, from one of the mosques in Morocco, and this is, for example, the pattern of a nano pillars, which looks really similar. So science imitating architecture, of course this is artificially made, so we see the architecture, we see something possible, and then we can also do it now in the lab. And this is one interesting example, just as it resembles. So how this molecular symmetry is, of course, more present in architecture. So architecture also benefits from science as much as we benefit from the architecture for inspiration. So today we can make invisible material, almost invisible and clever materials for architecture. We can have breathing houses, we can have CO2 recycling, we can have self-cleaning surfaces. Everything which is today used in architecture is made by science. And you know, this is the image of some of the architecture of the future. This is one of the clever materials that we designed, again, artistically enhanced for presentation. And I will close now. These are some nanomaterials that we like to, you know, whenever we scientists present our work, of course we need to draw attention to it, because we want to get funding. I'll finish in a minute. We want to get funding, and if you want to get funding, you also need to have appealing work. Something useful, but something which is also going to capture the imagination and the first instance. So for example, we like to play with our images as well. This is from Harvard University, where they grew these nano flowers. So these are self-assembly of molecular structures. This is all on the nanometer scale and done chemically. So, of course, this is enhanced then with a color. They add false color to blue, they add false color to create this almost alien structures to appeal a little bit more. We grew this nano flower completely by chance. It's not useful for anything, it just looks crazy. So we sometimes use it. This is made of DNA and gold. If you know what it's made of, it's really a little bit strange. So we call it a nano flower, or you can recognize all kinds of patterns in your images. And just to close up the answers to that question that I asked, how did being able to see down this atomic level change how we perceive nature? I believe a lot. You know, we can see the things we could never imagine we could see. And the more we see, the more we understand how little we understand. And there is also a wonderful mysteries yet to be discovered. Can we, inspired by molecular aesthetics, become more sensitized? I think we do. It's not destroying our aesthetical feelings. It can only help us to understand the nature, to appreciate it a little bit more. And some of the artists that I encountered also started using these materials also to build new stuff. And are we going to bring to life new art forms, change the architecture, create new nature? Probably yes. And it's very hard to say if this is good and bad. As you know, many things in the science, we would never dare to put them in the category. So finally, something for the end. One of the examples of collaboration between scientists and artists. I work quite often with Joe Davis because he's an artist, but he's also a very well-trained molecular biologist, self-learned molecular biologist. And so sometimes he comes with ideas, so he will send me the email. So now he had the idea of making a DNA airplanes, which he's going to fly and self-destroy. And just to you know, create a new artistic project. So he sends me this very scientific emails, like we need to use gyrase and topoisomerase, which is really molecular biology language. And I constantly ask him, how do you know that? How do you know that? And he said, I learned everything from the books and by asking questions. So he's probably better trained molecular biologist than I am. So we are now going to work with him, and we will do it in my lab and present it in Center for Art and Media next year. So this is an ongoing collaboration, and I wanted to tell you that this is really possible. I open my labs to the artists, they can go and play and observe my students. I also think this is very good for my students to get simply, you know, some strange questions or the questions they don't expect, and this forces them to think differently. Okay, with this I will close, and we also have a book out, and I wanted to invite you all to come and visit Globale and Renaissance 2.0, which is going to be taking place in 2015, and it will be dedicated to art meets science. So lots of scientists and artists will come and exhibit together. Okay, thank you very much for your patience with me, and I'm open to the questions.